Welcome to part 1 of the Central Dogma series, Nucleic Acids, the Building Blocks of Genes. We will discuss what makes up genes and the code behind creating them. Have you ever wondered why and how you have a different eye color than your friends? Maybe even how your sibling can have a different eye color than you. When we see plants like a petunia can come in many different shapes and colors, these physical characteristics are determined by genes. Genes are not the genes you wear. I'm talking about the basic unit of heredity passed from parent to offspring to determine expressed traits. That might be a mouthful right now, but we are going to dive into how this works. The term genes was introduced by Danish botanist, plant pathologist, and geneticist Wilhelm Johannessen in 1909. It was inspired by the ancient Greek word genos, meaning race, kind, and offspring. Where can we find genes? We see what they can do to a plant's size, shape, and color, but where are they? The amazing thing is genes are in every cell of an organism. If we take this tomato plant and zoom in on the leaf, then individual cells, and look at the nucleus of each cell, we will find strands of DNA containing thousands of genes. Let's investigate what DNA is. DNA stands for deoxyribonucleic acid. DNA is found in the nucleus of all eukaryotic cells and in the cytoplasm of prokaryotic cells. DNA contains and stores all genetic material. More specifically, DNA is the hereditary material in all living organisms, like trees and leaves, humans, pet dogs and cats, fungi like mushrooms, insects, even microscopic, single-celled prokaryotes such as bacteria that can cause human and plant diseases. The structure of DNA is a twisted double helix. If we untwist the DNA, it looks like a ladder. DNA is made up of 4 different building blocks called nucleotides: adenine, thymine, guanine, and cytosine. Each pair of nucleotides makes up a rung of DNA. For simplicity, we are going to assign specific colors and letters to each nucleotide. To construct the DNA structure that looks like a ladder, adenine and thymine always pair with each other with 2 hydrogen bonds. Cytosine and guanine always pair with each other with 3 hydrogen bonds. Remember A to T and G to C. Each hydrogen bond is weak, but there are a lot of them keeping the 2 strands of DNA together. Attached to each nucleotide, there is a sugar and phosphate group. The poles of the DNA ladder are connected by phosphodiester bonds between a sugar and a phosphate group of the neighboring nucleotide. Let's take a closer look at the sugar and phosphate groups. To build the phosphodiester bond, the oxygen of the 3' hydroxyl group of the sugar must bond with the phosphate group. Here we can see the highlighted groups that must break away to allow this bond to happen. 2 hydrogens and an oxygen are removed to form water, and a strong phosphodiester bond between the sugar and phosphate group is created. If we number the 5 carbons, we can see the 5th carbon is attached to the phosphate group, and the 3rd carbon is where the hydroxyl group was, but now has formed the phosphodiester bond. This designation will help us determine the 5' and the 3' orientation of DNA. Let's get back to making our strand of DNA. Now that we know how the phosphodiester bond forms, we can add more nucleotides to the 3' end of a strand of DNA. Don't forget the hydrogen bonds and the phosphodiester bonds. Now we've made a small strand of DNA. The 5' end is designated by the phosphate group, and the 3' end is designated by the hydroxyl group on the ribose sugar. Remember, this was determined previously when we numbered the carbons. Can you see how these ends are opposite of each other? This is because DNA is anti-parallel, meaning they run in opposite directions, kind of like a highway. One lane runs north and the other lane runs south. The chain of nucleotides we have constructed is called a nucleic acid. If you recall, DNA stands for deoxyribonucleic acid. Looking at this structure, we could be wondering how this looks like an unwound ladder. We can simply overlay a simplified structure of DNA. Doesn't this look so much simpler and more like a ladder? The nucleotides paired together are also called base pairs, and the amount of DNA varies between organisms, with bacteria typically having less DNA than their eukaryotic cousins. These base pairs are aligned in a specific order that makes up codes of thousands of genes. A gene is a segment of DNA that is used as a template to make a segment of RNA. Most RNA is then used as a template to make proteins, which affect the appearance or functions of an organism. This includes our eye color, and in plants, flower color like purple versus yellow, leaf shape and size like the large elephant ear leaf or the leaves of a Swiss cheese plant that has natural holes in it, as well as protection from pathogens and diseases, like we are seeing with this hypersensitive response on the leaf. But strands of DNA are huge, and if we wanted to move a whole strand of DNA, it would take an extreme amount of energy. In cells with a nucleus, eukaryotes, DNA is copied into smaller nucleic acid fragments called RNA and transported to the cytoplasm. RNA stands for ribonucleic acid and is in the cytoplasm of cells, and is the means by which plants and other eukaryotes transport genetic material out of the nucleus and into the cytoplasm in order to create proteins. If we unwind DNA's double helix, it looks like a ladder. RNA looks like DNA cut in half. RNA molecules are much smaller and easier to transport. RNA is copied in short segments to be transported efficiently from the nucleus to the cytoplasm. RNA is made of 4 nucleotides. Adenine, guanine, and cytosine remain the same, but thymine is replaced with uracil. A phosphodiester bond is made between sugar and phosphate just like DNA. Let's look at this bond formation again. RNA sugars have an additional hydroxyl group, unlike DNA, which only has one hydroxyl group. Here we can see the highlighted groups that must break away to allow this bond to happen. 2 hydrogens and an oxygen are removed to form water, and a phosphodiester bond between the sugar and the phosphate group is created. Where does the RNA get its code from? Here we have a DNA strand. The DNA strand splits and enzymes are used to copy the remaining strand. Remember, C and G are paired together, but since we are making RNA, A pairs with U. Now that we've copied the DNA, RNA can transport the genetic material. Remember, RNA copies portions of the DNA containing specific genes. This could be for flower color or leaf shape and size. The possibilities are endless. Let's look at this whole tomato plant. If we take cells from different parts of the plant, like the flower, tomato fruit, leaf, and stem, and look inside the nucleus, we will find all the plant parts contain DNA, and the DNA contain all the same genes. But the expression of certain genes is what makes these plant parts different. Flower genes will be expressed in the flower. Tomato fruit genes will be expressed in the tomato fruit. Genes specific to the leaf will be expressed in the leaves. And lastly, genes specific to the stems will be expressed in the stem. All cells have the same DNA and contain all the genes, but expression of the genes is different depending on where the cells are. Now that we have gone over DNA and RNA, let's summarize what we have learned. DNA stores genetic information and RNA transports genetic messages and enables these messages to be used. DNA is a double-stranded helix and RNA is single-stranded. The sugar groups of DNA have one hydroxyl group. That's how DNA got its name, deoxy. This means containing less oxygen, whereas RNA has 2 hydroxyl groups. The nucleotides of DNA are adenine, thymine, cytosine, and guanine. Adenine and thymine have 2 hydrogen bonds, and cytosine and guanine have 3 hydrogen bonds. For RNA, thymine is replaced with uracil. Now that we know the functions of DNA and RNA, we can explore the central dogma of molecular genetics. Central dogma starts with DNA. Since we have multiple cells containing DNA, the replication of DNA occurs. The process of DNA being copied to make RNA is called transcription. And lastly, once RNA has transported the code from the nucleus into the cytoplasm of the cell, translation occurs to create a functional product like a protein. The central dogma is the process by which DNA code is converted into functional products that can then build noticeable structures like the leaves, flowers, fruits, and stems of a tomato plant.